The overall goal of this technique is to monitor synaptic activity of visualized synaptic boutons at the Drosophila larval neuromuscular junction, an excellent model system readily amendable to genetic manipulations.
This technique can address key questions in neuroscience, such as how mutations of synaptic proteins regulate synaptic transmission. The main advantage of this technique is that the activity of a limited number of release sites can be monitored, and recorded currents have fast kinetics and can be easily resolved.
To begin the experiment, prepare the microelectrode puller. Use a microscope at 35x magnification to ensure that the inner diameter of the pulled electrode is in the range of 7 to 10 micrometers. Store the capillaries in tightly closed containers to prevent dust accumulation. Next, fire polish capillaries using a microforge. Ensure the final inner diameter of the polished electrode is 5 micrometers. Then, move on to bending. Fix the electrode in the manipulator and position the tip over the filament, not touching it. Set the heat value to 60 to 70% of the maximum and press the pedal of the microforge for 1 to 2 seconds to heat the filament. Use an L-shaped needle to gently pull down the tip of the electrode and make the bend at approximately 90 degrees. Hold the electrode over the flame of the torch by hand and make the second bend at 7 to 10 millimeters from the first bend and at an angle of approximately 120 degrees. Prepare hemolymph-like or HL3 solution.